All right, perfect. Well, thanks to all the organizers for inviting me to this very interesting conference. My talk will provide an overview of a very broad picture of cognition, as you will see, that intends to be unifying by looking at diverse kinds of cognition that we observe in natural and artificial systems, and see whether or not we are able to perform, to construct a theory of cognition beyond the standard neural network picture. All right, the origins of this come from a workshop I co-organized at the Santa Fe Institute along with David Wolpert in 2014, related to the major evolutionary transitions, and in particular the possibility. By major transitions, I mean the big innovation events that have happened in evolution across the history of our planet, meaning the emergence of life, of genetic code, cells, multicellularity, et cetera, all the way up to language or consciousness. And the possibility that we can actually think in terms of synthetic alternatives, meaning that nowadays in the lab, we can actually mimic the evolution, for example, of language in evolving robots. We can build, as I'll show you in some examples, use synthetic biology. We can use some complex alternatives to evolutionary innovations, use artificial life and statistical physics. And as an outcome of that meeting came out the idea of what about those transitions involving the emergence of cognition? And we did another workshop that I'll talk about a bit later, involving precisely this idea, the idea of what about the big transitions in evolution that have to do with cognitive innovations. The first meeting eventually ended up in this special issue in Philosophical Transactions, which covers a number of these different systems. I very much recommend it to you. And we explore here a number of things from, as I was saying before, protocells, symbiotic systems, synthetic alternatives to viruses, et cetera, et cetera. And on top of that, many relevant questions emerge that connect evolutionary thinking and thinking from the mathematical model, computation theory, and statistical physics. There's some of them are here. I'm going to go through some of them, but just would like to highlight the relevance that seems to emerge from the analysis of all these phenomena in terms of unifying principles that might have to do with, in particular, with phase transitions. And as I was saying, one of the very important innovations that you observe in evolution is the emergence of cognitive complexity. Extremely important because that means it represents a step beyond genetic information, genetic memory, et cetera, et cetera. With the emergence of what we call usually cognitive agents, we start to be able to interact with the environment in totally adaptive ways that scale in a time scale that has to do with the lifespan of individuals. This is one particular paper that I like very much about the evolution of information connected to these major innovations by Eva Jablonka here in the picture and Marion Lamb, where they stress this particular thing, that innovations are not always to do in the evolutionary picture with genes, that we have to actually pay more attention into emerging factors that come out when non-genetic information is in place, all right? In relation to cognition, it's a lot of open questions, as you know very well, right? Why actually brains? Why brain complexity? Why cognitive complexity, which is costly? Why this is there instead of a biosphere made just of microbes, which are simple replicators, and that's all. And this seems to be directly connected with the problem of prediction, which I'm going to mention. If you are able to foresee the future, you're able to predict. That kind of cognitive complexity, that ability to reduce the uncertainty of the environment automatically provides a source of fitness, right? Beyond the fitness of pure replicators. And that goes all the way up into what makes us different cognitively, and open questions about the nature, for example, of consciousness. And in particular, why brains? I was saying, why brains, and even how do you define brains? Well, I want to highlight two ideas. One that is around what's the engine that propels the creation of brains in evolution. One is the moving hypothesis. It's this idea that Ginas explains very well, that if you have to move in space, so you are an embodied agent, you have to move in space, searching around in an uncertain environment when you have to detect things and respond to challenges, having a brain is something that is extremely important. You have to move a body, but you have to integrate information, and you have to integrate information in a reliable way. And the other thing, the idea that has been around in different talks here, is the prediction. Brains are prediction machines. And as I was saying before, prediction provides a big component to fitness. On top of that, it's also something that is very important when we discuss about the evolutionary origins of things. One relevant question here is, what's the role played by contingency, and if you want, random events through evolutionary history, in respect with strong constraints that seem to be imposed by a number of features that you observe in systems that have embodiment. On the one hand, that's the perspective taken by Stephen Jay Gould at left, is the idea that if we were able to kind of rerun the tape of evolution and repeat the whole process, we will end up in a biosphere that will be completely different, completely alien biosphere that will include the cognitive agents, right? Whereas other people, and I'm on that list, particularly people with this complex systems thinking, we think that actually there are so deep constraints into what is possible in evolution that if you rerun the tape of evolution, you will observe a different biosphere, but very familiar, all right? And this book, Life Solution, is a kind of a compilation of ideas that have to do with this idea of convergence, that despite the different historical trajectories that lead you into innovations, the kind of innovations that you can eventually have has a very limited repertoire. I put the example of the eyes here because, for example, the eye that we carry out as humans is a complex camera eye, has been reinvented a large number of times independently in evolution, in very different groups, in vertebrates, but also in invertebrates, even in jellyfish. And there's another example I'll show you later that is quite puzzling. And essentially it's the same design, again and again, repeated many times. And even in groups like invertebrates, I put the octopuses here because, you know, it's a very hot topic now about the complex minds of these organisms. They are invertebrates. They achieve their brains totally independently in evolutionary history from vertebrates. And yet you end up in a design where you actually detect very well with a simple eye inspection the structure of a cortex, the complex neurons that we know. There's a rearrangement of things here because they have these eight arms, each one having a kind of a little brain. But essentially, you end up with that, and also the complex eyes I was mentioning before. Okay, going to cognition, one of the big issues that we have, if we look at comparing different kinds of animals, is precisely definitions. How do you define, for example, intelligence? I'm not going to give you a closed answer to that because I think that is still a debated issue. You will find many papers talking about intelligence, right? But it's as you can imagine, it's a controversial issue, right? As when we talk about intelligent houses, what's the meaning of that? Well, if they open the lights automatically when you enter, that's not a terribly big intelligence, right? So defining intelligence is a big challenge. It's one paper that makes a big attempt in this direction. I think it's limited in the way it tries to actually end up with a formal definition, which is very much in the machine learning picture, right? This is a definition where you have a performance that actually weights the amount of intelligence in a set of environments, which the sum is done on that, all right? And Nevertheless, I think it's an interesting attempt of the kind of things that should be tried in the future. But anyway, if you want to actually think in intelligence, cognition, in a very general way, taking into account all the possible ways you observe cognition in nature, it's important to try to build a space of possible cognitions. And that brings into the follow-up of that meeting that was mentioned before. We eventually organized another meeting. I organized this with Melanie Moses from University of New Mexico and Stephanie Forrest, that is now at Arizona State University, that we label precisely liquid brains, solid brains, right? Where actually the idea was precisely to bring together scholars from very different disciplines, working from single cell organisms to artificial intelligence to ant colonies, immune systems, microbiomes, etc., and see whether or not we can end up in some kind of general picture. Well, what was this solid and liquid brain, right? As usual, when you try to make a kind of big definition, you are limited by a number of constraints again. But when I say solid, I'm referring to brain structures which are the standard ones, meaning that you have neurons that are, once development finishes, are located in given spatial locations and are connected to other neurons in a more or less stable way. We know that weights change, right? But allows essentially to define static connectum, right? For systems like a hydra, which is a very simple invertebrate that has this diffuse, semi-regular, random, homogeneous, if you want, neural network. You have a simple connectum, whereas C. elegans has a kind of a more modular structure, and you have heard about brain considerably. So what is the meaning here for liquid? Liquid means that you have agents that can be more or less complex, that carry information, can process information, and especially connect to other agents in the system, and that by contrast with the solid brain picture, move around. So you don't have stable connections over time. And colonies will be an example. The immune system will be another example. And I'll try to go through several limit cases to see. Termites, for example, are another group, and it's nice to look at that because, social insects are one of the best successful groups in the ecology of our planet. They have transformed the planet, and they have this collective intelligence that cannot be reduced to the understanding of single individuals, right? That's actually a great example of what a complex system is. And they build complexity by using self-organization. So interactions between individuals and the scaffolds that define their nests, which are dynamical systems, allow actually to bring something that defines a superstructure, right? Kind of a superorganism, and that's at that level where you actually can talk about cognition. I put the equations here because they actually, one of the things that do this kind of organisms is build large-scale structures that can be sometimes 1,000 times larger than the individuals, and that Turing-like phenomena allow actually to explain the mechanism by which this happens. Okay, if you go into and this is a material that you know well, into the description of solid brains, the modeling of that, of course, neural networks, as we use all the time, it's the standard. I put also here genetic networks, and Stu Kaufman here, because genetic networks, as defined right at the beginning, were actually very much close, and they are very much close in many ways, to the kind of modeling that we use for neural networks, right? And I don't think that this is actually a random observation. So by defining solid brains with neural networks, we use our formal kind of definition. We have the attractor picture, where essentially, as you know well, we live in a multidimensional space where everything that is interesting is in the connectivity matrix, right? And that connectivity matrix changes through learning, but essentially is what it makes things interesting and relevant, right? I put Hebb's rule as defined in Hopfield's model because kind of one of the standards, but it's going to be interesting to discuss later on. And here, memories are kind of the central topics here. The possibility of storing a big amount of information and recovering that information from incomplete data. The other part of the story that has been also discussed by several lectures here is dynamics, right? Beyond the attractor picture where you end up in this attractor state and you remain there, of course, the real brain is a highly dynamical system. And we also know that everything seems to point out in the direction that it is actually not any kind of complex dynamics. It's very much clear that it lives somehow into this critical state, all right? And that, in terms of modeling, is connected very often, and it's an important connection to make here, to these kind of transitions, which are epidemic-like dynamics with an absorbing phase. And I point to that because that has been used extensively. Extensions of that, of course, is a very simple model approach. And I'm going to go back to that when I discuss how to model liquid brains. And of course,
density of a rice grain is smaller than the mass density
of course, is the idea
One could
of,
therefore expect inertial effects to be of less significance to rice
there's
grain
the,
dynamics.
I forgot to remove the volume. But anyway,
in 2000,
there's the metaphor of self-organized criticality. How do you self-tune yourself into that? Well, what kind of liquid systems do we want to discuss? And I'm not going to limit myself to this liquid system, but go also to other extreme cases that are relevant. Examples. Well, ant colonies are a clear example that has been under a lot of scrutiny in terms of theoretical work. The immune system also. I'm not going to spend time in the immune system, just to mention that this is one of those complex systems that are extremely interesting for many reasons that are relevant here for one very good reason. The immune system is a system that is actually is embodied within ourselves, that works in such a way that essentially has to detect self and non-self. So it has to detect who is not part of you and who is part of you. And that has a lot of derivatives. And modeling the immune system, actually, for the people who actually went into looking at groups of cells that have the specific ways of finding out antigens from pathogens that invade our organism and that interact with them, and did that with models that are not very different from neural networks. And I put this particular paper by Giorgio Parisi. It's an old paper in PNAs, where actually he makes the analogies between the kind of the Hopfield spin glass-like modeling in the immune system. My point here, the only point here is that this is a liquid system. Cells move within our bodies in the bloodstream, and they interact under ways that you can actually define it with a weight matrix. And more importantly, they display memory, they display learning, right? And so they are adaptive, although with a big limitation, which is essentially, as I was saying, the function is detecting self from non-self and responding to the non-self challenges. Going to cells, because that could be the minimal kind of system that we can consider. And cells, within cells, we see a very dynamical state, right? Which is not exactly liquid, because it has a number of really interesting phases, and we know more and more every single day. But one interesting thing that I think is worth mentioning is that if we want to go into the cognition computation picture, one of the interesting things we find in cells is that actually we can identify very well some kind of machinery that has to do with computations. What's the meaning of that? Since the beginning of molecular biology, which started also almost at the same time as information technology, the ideas and the jargon from information theory immediately went into molecular biology. So we talk about translation, transduction, transcription, coding, etc., etc. And an amazing thing that I just want to point out, because I think it's relevant to think for what it says about the limitations of the possibilities that we can find out in natural systems, is that Alan Turing, when he describes the now famous Turing machine, in order to make a general model of computation, he ends up in talking about a long tape, right, a very long tape that is being read by a machine, the head of a machine, right, with internal states. And by reading that tape, you actually perform a given function, right? There's a number of issues here about what is the meaning of that. But it was surprising because that when Turing wrote his paper, it was years ahead molecular biology. Before molecular biology actually established the existence actually of machines that read long tapes. We call it DNA, for example. It can also be RNA to make functions, right? And having stop and initiation parts is only one part of the story. But if you go into other machines, like, for example, ribosomes, which are very complex machines that read RNA molecules to create proteins, you can actually show that the way this works to a very large extent is a finite automaton machine, all right? But it's more than that, because if you actually look at cells, we know now that cells can have memory, can learn, right? Of course, there's a big question of how far they can go with that, right? But you have seen movies of cells that are predators. You will have seen that they can actually have a lot of complex behavior. And one of the things you can actually see when you look inside the networks that connect, for example, proteins that move information from the membrane to the inside world, you see these complex cascades that very much reminds us of feed-forward neural networks. Dennis Bray, in these papers and follow-ups that he did, actually goes into this kind of analogy in a deeper way. And as I was saying, the fact that cells move, remember the moving hypothesis, they have to search in their environments, they have to do it in a rather sophisticated way, sometimes can be very much proactive in the ways they move. I want to point out that actually movement in single cells, like the one it's shown here in these three snapshots, happens thanks to a very interesting machinery that inside cells, the microtubules, which are extremely dynamical systems that actually live close to the phase transition between order and disorder, right? And that's not a chance even. Movement itself, for example, for bacteria like E. coli, is controlled by a system that very much remembers, again, a feed-forward network, where you have detectors that sense an environment that can have positive and negative signals. It's integrated in a module, this chi, y, zeta, etc., that actually makes decisions about whether or not you move the flagella. So you change your moving behavior, all right? So you can actually think and talk about some bacterial intelligence. What about criticality? Because we mentioned criticality is one of the potentially very, very important pieces of complex cognition, right? Criticality in cells has been around since a long time ago, when Kaufman suggested the idea that genes are connected within cells in a phase transition boundary between order and disorder. And in relation to that, there's been a lot of interesting work, but it's not exactly what we're talking about. It's not exactly the idea of dynamical criticality that we will be interested in talking about cognition. So as I was mentioned before, one avenue to explore innovations, evolutionary transitions, is to actually create them in the lab using engineering tools. That's one of the things that is done in my lab. We do theory, and we have a wet lab of synthetic biology, where we use all these tools of this discipline that's been exponentially growing in the last 20 years, using genetic engineering to actually create new phenomena within cells, right? Things that cells don't do, we actually try to make them happen. We just published last week a paper in Nature Communications, where we actually show that, for example, self-organized criticality can be engineered within living cells. Let me explain that, because it sounds like an oxymoron, because self-organized criticality by definition, as you know, means that you organize yourself into the critical point, right? In the picture of the second order transitions I mentioned at the beginning, with this absorbing phase, you actually move into that direction, and you take the picture of the metaphor of the sand avalanche. You have a constant push into increasing the number of grains of sand, eventually you may grow the angle of repose of the pile, which can only go into a maximum level, the critical angle, and then you have this dynamical set of avalanches. The interpretation of that is that there's an interaction between the order parameter, which is the activity of the system, the avalanche size, and the control parameter here, which is the angle of repose of the pile. And we implemented that using a genetic circuit. This is the one on the bottom left, right? We essentially, to make the long story short, we have a gene that is expressing constantly a given protein. We have another gene, that's in the lower part, that expresses another protein. Both are destroyed by a machinery that is there in the cells. That was called the proteolytic machinery. But the second gene that gives a protein, this protein, if the destruction made by the proteolytic machinery is not efficient enough, you will have accumulation of this second protein. And when you have accumulation, and this follows a threshold dynamics, they form dimers, and the dimers repress the first gene. Our first gene makes a protein that is our order parameter. The second gene makes a protein that is our control parameter, right? You can actually build this in terms of a system of differential equations, the mean field theory that is shown in the upper right here. But essentially the idea is that the interaction between activity in the system and detection that the activity is reaching a threshold, and that in some way this triggers a feedback, a negative feedback that reduces the production of the first, right? It's completed here, right? We were able to show that the model rightly predicts self-organized criticality. It's engineered, so we engineered essentially this motif of order and control parameters interacting, so that to facilitate the possibility of having a critical state, except that we need to actually have a minimum level of processing of our protein, of expression. Otherwise, it doesn't happen, right? This is confirmed by the experiments, right? I think it's a very neat example of how genetic engineering and synthetic biology can actually help constructing new phenomena. And we conjecture, and that's kind of the follow-up that we want to do in our lab, that if we were able to actually connect, that's the lower part of the diagram, connect our, we call it self-organized critical motif, connect with, for example, the movement of the flagella, we could actually engineer behavior in bacteria, okay? And that's one of the ideas we have in mind. And just wanted to mention something that I found incredibly amazing about the complexity of cognition you can actually find even at the single cell organisms. Remember I was mentioning that our camera eye is shared by a number of different species that has been independently invented in very different parts of the evolutionary tree. A very surprising finding some years ago is that there are some single cell organisms, right? Unicellular organisms in the sea actually, that have this kind of camera eyes. They are formed by symbiotic bacteria that aggregate, forming a retina, forming a crystalline. It's kind of a really amazing thing. Clearly they use that for something, but because they are a bit rare, it's still not well-known, characterized, but you can see how complexity can actually be found in the most unexpected places. Another organism that belongs to our hierarchy of fluid, liquid cognition or networks or brains is Physarum. Physarum is a single cell organism, is the largest single cell organism you can find in nature. Right? You see in the upper left, it's a petri dish. This is a single cell, believe it or not. It has lots of nuclei inside, but essentially it's a cell that can expand and deform in an extraordinary way to search its environment. And it's suitable for a lot of experimental work, right? For example, this is a classical work in the movie where you use a labyrinth. You kind of put Physarum sampling in everywhere, and they like to eat, for example, cornflakes. So you put one at the beginning of the labyrinth and another at the exit, right? And originally, the fungi, it fuses itself, it just keeps moving. It forms these tubes that are extremely dynamical and pulsatile, so you can actually measure kind of electrical signals moving around. And it self-organizes in a way that it detects the presence of these food sources, right? And it just optimizes those parts of the system that make the transfer of food as efficient as possible, right? And it ends up in a single tube, right? The other tubes are just it's kind of a selection process. They just destroyed, and you end up in the solution of the labyrinth, right? You can actually build a lot of interesting experiments with Physarum, including learning between two solutions in a symmetric two choices path, right? Where you have a good and a bad choice, and actually model that using simple mathematical models that essentially capture these dynamics of tube formation. This would be here, if we're in space, the connectivity matrix that you have, and except that now what is computed and how this is computed, how do you know that computation has been fulfilled, is in the form. The shape of the final Physarum configuration is the computational solution to the original problem, right? It has been extensively used in many, many different ways. You can build logic gates with Physarum. You can solve mazes. It has been recently used to actually mimic, kind of simulate the large-scale structure of dark matter in the universe, right? But a very important point to be made here, that suggests that Physarum is kind of very complex in terms of cognition. But in fact, Physarum solves problems that have to do with a kind of minimal path problem, right? Kind of least action. Once the boundary conditions are well established, right? And that's why you can make a lot of different experiments to show that Physarum can solve topological problems, all kinds of problems. Physarum essentially solves this least action, right? So that means that if you want to think Physarum in the wild as a very complex organism, maybe we should be careful in saying that, right? Okay, that connects with ant colonies. What kind of things can be solved by ant colonies, right? Well, first of all, a reminder that ant colonies are made by sometimes huge amounts of ants. Not always. There are colonies that are small, while other colonies have millions. And I'll go into that at the end to think about what's the trade-off here between the individual and the collective complexity. What kind of problems can they solve? They can sense their environments in extremely flexible ways. And how they do that? Because single individuals are not likely to be able to sense the environment as a distributed kind of signals. So it is the colony who does that with the help of what? Usually with pheromones, with signals, chemical signals that define an external field, right? That they can create and reinforce. And that because it evaporates, just part of the simple decay, right, is a dynamical system in itself. And for example, ant colonies can solve problems like finding out the shortest path, right? When they are confronted with two paths along in a short path, separating, for example, the nest in one side and a source, a food source in the other part, right? Finding out the shortest path is a good strategy because it optimizes resources. How they do that? Well, at the beginning, they go randomly in both paths, right? And when an ant finds out a food source somewhere, when it goes back, it leaves a little mark, right? Kind of marking a chemical signal that success has been happening there. At the beginning, you will have marks in both sides, right? But the longer path will imply that the marking, the pheromone, is going to be dissipated also more quickly because you have actually more surface, right? So essentially what happens is that the colony creates something that they sense, right? They amplify. And the amplification phenomenon and the fact that, you know, the shortest path will be more amplified creates a process of amplification that actually can be interpreted as solving the shortest path, right? In many colonies, we can actually do that with a symmetric reach, right? Exactly, it's the same paths. and where you will see actually symmetry breaking, right? That actually the amplification phenomenon is there. This can be modeled in a very straightforward way. There's a lot of literature on that, and the evidence that those colonies that actually use noise to actually introduce some randomness in decisions can actually make more efficient the way they find their solutions. We are also exploring in the lab this problem. We want to implement this with our someday microbial ants solving shortest paths. But let me go into this other problem that was explored years ago, and we are re-exploring nowadays, of ant colonies that have the following. You have heard about that. You have ant colonies where you have different tasks that have to be implemented. Sometimes these are implemented by kinds of ants that have different shapes. They're morphologically different, but in actually the majority of species are monomorphic. So essentially the ants have exactly the same body, but they have to make decisions, which means that inside their brains, somehow they have to be states that allow to make decisions about what to do. Nest maintenance, foraging, patrolling. There are several things you can do. Deborah Gordon, who is one of the best known experts in the world on ant colonies, explored that in the field. She's still working on that and characterized very well the amount of workers doing different tasks and how they switch from one task to the other. And the beautiful thing of this is that what you can actually detect is that in the space of possibilities, where you can have these 4 different tasks, you can actually define them as states, right? And make a very simple model inspired in the real observations that maps very well into a neural network where every ant can be considered as a multi-spin system, and that you can define a matrix where, depending on with whom are you interacting, and in fact, in nature, how many times you have interacted is very important, right? Makes you decide to shift, right? In such a way that given some requirements you have in the colony can adapt into different configurations. The interesting result from this mathematical modeling, which is kind of, as you see, statistical physics approach. What are the attractors here? Again, you have a liquid system, and attractors are not in a connectivity matrix. The attractors here are numbers, the numbers of ants doing particular functions, right? Very different from what we are thinking about usually when we think in the solid brain picture, right? That, I think, gives you a glimpse of the limitations that might be implicit in liquid cognition, right? Which is something that we don't have a theory yet about what kind of the complexity can be achieved by these liquid systems. On a different issue, but again connected with ant colonies and dynamical states in criticality, some work that we did some years ago involves a different phenomenon that we see in another class genera of ants. In these colonies, which are small, right? You can have it in the office. I had it for a while, one of them. You can actually show that, you can actually see that individuals, from time to time, they do nothing, right? It's kind of far away from the standard metaphor of ants as hard workers. They can be totally immobile, as in the picture here. And then you can see an ant starts to move and then touches another ant, and that ant starts to move. And eventually you have kind of a wave of activity, right? The waves are not completely ordered, right? They're kind of a interesting compromise between order and disorder. And when they are active, they clean the larvae, they go for food, etc., all right? Well, that was kind of a puzzle. We explored this problem using a kind of, again, a fluid neural network, meaning that now our ants are only one spin state, right? Active or inactive. And we redefined a little bit the standard rules of excitation, but in the end, as you can see, it's not very different from the models we use in neuroscience to model propagation of activity in neural masses, right? And now the thing is that depending on your state, will interact in a way or another, but it's a simple matrix that allows that to happen. And what we find is that there's actually this Our individuals are not oscillators, so it's not just coupling oscillators having synchronization, and instead there's this kind of emergent phenomenon that ends up in a synchronized activity. And interestingly, we know that happens in the real world. In these ant colonies, you have a critical density of ants, right? And if you modify the ant colony, they form these circles of grains, right? So it's not rigid. If you make, for example, density to be higher, they reorganize everything and go to this density that seems to be a critical density. Because what we find out actually is that it's this open to density. Essentially, you have the most complex kind of dynamical state, right? And just to point out that here, there's an interpretation that is functional, because you could argue, why not an ant colony where essentially everyone activates or deactivates at random? You have the same average activity level, but you can actually show, and that comes from computing theory, that synchronized activity here guarantees that everything that has to be managed in the colony, in this spatial distribution of objects, will be optimized. You are not going to miss anything. Whereas if you have the other strategy, you randomly activate or deactivate, you can actually calculate that some items in the system, like larvae or eggs or whatever, will be unattended, right? So it makes sense to be critical. We are also trying to do that in the lab with synthetic biology. I'm not going to give into details to that. And the thing is, right, I have shown you some examples. There are a few other things I want to mention at the end. This defines something that goes beyond the pure, let's say, the standard metaphor, right? The neural systems that we know very well, right? Or at least quite well. So one thing that we have been exploring is this idea of a space, kind of a morphospace with Olaf Sporns. We have a paper actually on the network morphospaces. We locate here 3 axes. The idea is to collocate in this space the possible that we observe, right? One axis is physical state, right? This horizontal axis, we have liquid and solid. Another axis is cognitive complexity. What's the complexity of decision making, if you just make simple decisions, or the capacity for having complex decisions that have to do with learning and memory, et cetera, et cetera. And in the vertical axis, developmental complexity, meaning that whether or not you have to go through a process that means that maybe starting from one cell or from one queen, if it's an ant colony, you actually have to build the whole system, right? In this space, we can identify easily some well-defined limit cases, right? For example, neural systems in the upper right corner. If you go to the left in the same corner, but the other part, in the liquid part, you have the immune system, right? A complex, diverse system that also is created by development. Whereas other things that can be created, like printed organs, are just in the lower corners of this diagram. And an interesting thing that we found, you always have Physarum and insect colonies, is that there's a big space in this space of the possible. And that's one of the beautiful and interesting things of building morphospaces. I want to point out the location of these systems is qualitative, is relative to others. We still don't have good measures to organize this in a quantitative diagram. But nevertheless, one of the interesting things of morphospaces is that sometimes you observe these empty domains, right? Which means that either it's not possible for evolution to explore this domain. It could be the case that there are big constraints that have to do with impossible physical states, right? Anyways, that's an interesting thing to think about. What is missing there? And for us, that we also do synthetic biology and try to make synthetic alternatives, is the question of, could we actually build something that is in that place, right? So just let me show you some of these systems, organs, standard organs, for example, that can be more reactive or less reactive, are also parts of this space. This is the whole range of possibilities that are allowed by bioengineering organs that can be what we call the organoids. These are they are kind of what would be the right word, kind of metaphors, if you want, of real organs that are built from stem cells, right? These are mini systems. That includes, for example, mini brains that have been also explored by several people working in network neuroscience that have limited capacities, but nevertheless allowed to do a lot of things in terms of biomedical research. The immune system, as I was mentioning, you can define here networks on different scales. And as I was saying before, right, they involve this high diversity, learning, memory, etc., etc. Of course, insect colonies, you can realize that I put here insect colonies between liquid and solid, because actually they are a combination of that. They have, on the one hand, the nest structure for the vast majority of social insects that are social insects have nests, right? It's a scaffold that is required for colony survival, right? With direct interaction with individuals, right? Which define a liquid phase, right? So in a way, they are in the middle between both possibilities. The microbiome, for those of you who don't know about that, the microbiome is something that is kind of a revolution within our understanding of biology and probably cognition, because we carry a microbiome inside us, has been co-evolving with us for millions of years. It's not just microbes that are there. You have around 4 of these cells for every cell in your body, and they seem to be pretty much decision-making systems connected with the immune system and connected with the cognitive system that we call the brain. So there's a whole hierarchy of things in terms of cognitive complexity that we need to understand, right? And they are also in the between as liquid and solid phases. Physarum, as I was mentioning before, again, is not clearly a solid system. It's not either liquid, right? And just to mention that one of the things that I was saying before is that the empty spaces may be a place where we can actually engineer complexity. That's a big challenge that we are considering these days. What other questions? I already opened some questions, I guess. Open questions that are there about the complexity and the diversity of cognitive systems that we see. Cognition sometimes can involve a brain, but the brain doesn't need to be a stable structure. And that happens, for example, it's kind of a puzzle here also, metamorphosis. In metamorphosis, you have one phase, right? Where you have the caterpillar that has a brain, it lives a life, eating leaves, etc. It experiences metamorphosis, and in metamorphosis, everything is destroyed, right? Everything is totally reshaped, including the brain. The brain is just gone, right? And it is reconstructed in the adult, right? These kind of reconfigurations happen in different systems and makes you think about what is actually the selective value of some things, right? In other species, for example, and that connects with the moving hypothesis, in ascidians, for example, you have a larvae that has a little neural center that is helpful while it moves around, right? Until at some point it settles down somewhere, and as the adult is created, which is attached to the substrate, the brain is digested. So the brain disappears and it's gone, right? I think it was Ginas himself who said that seems what happens with some professors when they become professors, right? I don't know, that's a bold statement. All right, things about complexity of the individuals. We just went very quickly into that, thinking in models of ants that are active or inactive, or have a small string that defines a repertoire of possible states, right? Like tasks, for example. But we have gone quite quickly about that because in a way they have brains. They have brains with millions of neurons inside their heads, right? So what about individual complexity? What happens with that, right? It's not a trivial question at all, right? During the development of each individual, right, is a construction of a brain. There's a number of things that we have been learning about social insects, like wasps that recognize the face of others, right? Is non-trivial what it's possible there, right? But what is difficult to actually assess, and that's an open question, I think is one of the most fascinating questions, is about the trade-off between individual complexity and collective complexity, right? And one of the things that we don't see in natural systems in ant colonies is, so to speak, brainy ants with big brains, right? We do observe something that is interesting though. In very small colonies, like colonies with can have 50 or 100 individuals that are very large, right? I saw them in a rainforest in Panama years ago. They are pretty much independent in a way. You see them going around and more or less making decisions by themselves. Whereas in very large colonies of actually millions of individuals, individuals are typically blind. They just make very simple decisions depending on who is there in the neighborhood, very simple chemical signals, and yet the collective does really amazing and complex decisions, right? As if there was a trade-off between the collective complexity and the individual complexity, right? This connected with a very interesting problem that is, as I was saying, it was kind of unsolved. In this paper by Dan McShee, he kind of explores this, the complexity drain hypothesis, right? That there's a trade-off between what an individual can achieve and what the collective can achieve in terms of cognition. And years ago, with my colleague Jordi Delgado, we tried to actually formalize that using coupled map lattices and special formal analysis of computation, the so-called epsilon machines that were created by Jim Crutchfield, and try to actually address that in a formal way. But as I was saying, this is still yet an open question. What about plants? And I bring that because it's a controversial issue. It's been in the last years very easily said that plants are intelligent in some way, right? And some people actually completely makes an abuse of language, in my opinion, saying that plants are intelligent, right? If you actually look at plants carefully, it's true that they can exhibit some kind of memory, exhibit some kind of learning, although the examples are very little. They do cooperate, right? But every single thing I said now can be done by bacteria, right? So there's a bacterial intelligence, and my suggestion is that maybe this plant intelligence is not so far from intelligence from other much more simple organisms, right? And that's of course directly connected with the fact that they don't have brains and they don't have neurons, right? And they don't move, right? That's an important issue. Remember, we actually said that the moving hypothesis is one of the ideas that is there, kind of suggesting that moving in an uncertain environment actually is what pushes the development of cognitive complexity. One little model we did, and we still have to complete and send to publication, is with my colleague Salva Duran, is what if you make a model of agents where you have a set of resources distributed in space, plus a resource that comes from somewhere, kind of the energy that comes from light, right? Which you don't have to search for. And if you stay in the same place, right? And that's our original condition, the initial condition, you can gather that energy. You are not going to exploit the other energy that is around and fluctuates, etc., right? But could be very rich. And so we can evolve our agents. And you allow agents to evolve, okay? What you see is that if resources are common enough, right? So if it is worth to spend energy because it's a cost, energy and have perception to move around, right? You can actually see that there's a kind of a phylogenetic tree with two main choices. You either are a plant and you remain, yes, sessile without moving, taking the energy from the external environment, or you become a moving agent, right? Which has this limited but present cognitive capacity of detecting what is around you, right? So our model, which is kind of a first attempt of exploring that, and I know there's a lot of limitations of the approximation, suggests this idea that if you don't have to move and you can exploit a source that you have to search for, you are not going to be a cognitive agent, right? And let me point out, just to finish, that it's actually interesting to see that there's a part of the plant life that involves decision-making, understanding the nature of what's going on in the environment, that does make that, right? That has this switching decisions, etc. These are the seeds, the moving part of the plant. They do have to make decisions, sometimes non-trivial decisions, right? Interesting, because that means that because you have to move, right, there's some cognition there that is necessary. And that would be all. I mean, I know it's a lot of stuff. You can see now why I have been not going into much details, but as you can see, I think it's a fascinating problem, full of interesting questions. And I must say that I think also that for the people as it's searching for new ideas within deep learning and other domains of artificial cognition, if you want to say that, right? I think that looking at these different ways in which nature performs computations and has capacity for making decisions and predict could be really interesting to actually maybe searching for other breakthroughs. And that will be all. Thank you.